Hi friends, it's Dr. Tim and today we'll be discussing a very exciting topic under biochemistry, enzymes. So before we continue, I'd like those who haven't subscribed to this channel to actually go do so now. Okay, I'm assuming you've done that. So enzymes, okay, before we go on, there was a video on introduction to biochemistry, and we define biochemistry as what? The chemistry of life, because that's the literal definition, right? Bio means life, so it's the chemistry of life. But for a more comprehensive definition, we say that biochemistry was the study of life at the molecular level. Okay, so if you are studying any living organism at the molecular level, what you are doing is biochemistry. So we say that there are different molecules in the body. There are major ones like your lipids, your carbohydrates, your proteins, your nucleic acids. These are major molecules in the body, and you also have water, which is the most abundant actually. So apart from those, those major molecules in the body, you also have other molecules in the body like your enzymes, your hormones, your organic pigments, etc. So for this video today, we'll just discuss enzymes because they're very important, right? Next we discuss is the characteristics of enzymes. So the first thing you should know about enzymes is that they have a high molecular weight. So all enzymes have a very high molecular weight. That's number one. Second thing you should know is that most enzymes are proteins. Not all, but almost all enzymes are proteins. Over 90% of them are proteins. That's two. Third thing that you should know is that enzymes are organic catalysts. I think that's obvious. It's in the definition, right? So if it's not a catalyst, enzyme, it's not an enzyme. So that's very important to include as well. So the fourth thing would be that enzymes are not used up at the end of the reaction. So that means they will go into this reaction as they are and they will leave as they are. So they'll just enter, speed up the reaction, but they will not be changed. They will not, undergo any change whatsoever. Right. So the fifth thing would be that enzymes are specific in their action. So enzyme A has a specific action on this particular substrate. So it will not act on any other substrates. You get that's how they work. So they are specific in their action. And the sixth thing will be that enzymes can be regulated. So enzymes can be regulated. All right, so you might be wondering how are enzymes regulated in the body. So the body has different ways of regulating its enzymes. All right, the first one is called feedback or allosteric inhibition. What that means is usually when an enzyme acts, it acts on its substrate and then it gives rise to some product. So if that product is in a high amount, that high level of products can inhibit the enzyme activity. So that's called allosteric inhibition or negative feedback. So that's the first way. Second way is that the body has some functional proteins. So and some of them are regulatory proteins. That's the common way now. Regulatory proteins number two. So regulatory proteins actually help to regulate enzyme activity. And the third one is called covalent modification, when you may probably add a phosphate group to an enzyme and then the change in structure causes the enzyme not to function again because there's this structure function relationship in the body. Okay. And the last one is something called proteolytic activation. This one is particularly for protein digesting enzymes. That's enzymes that will digest proteins. Okay, so they are secreted in their inactive form. It's called zymogen form. So the reason why they are secreted in that form and they're activated by something else is because for those enzymes that digest proteins, you don't want, because the body itself is made up of proteins. So for example, the stomach cell that produces enzymes that digest proteins, they secrete their enzymes in inactive form. Why? Because the stomach itself is made up of what? Proteins. The cells, the stomach are proteinous. So if they are inactive, if they are in an active form initially, they can digest up the stomach. So the point there is for proteolytic activation is for enzymes that will digest proteins, right? And they are secreted in what? Inactive forms or zymogen forms, and then are now activated when there is there is need. Yeah, so that's it for regulation. There is negative feedback or allosteric inhibition. There is regulatory proteins. There is covalent modification, and there is proteolytic activation. And yes, very important, you need to know the mechanism of action of enzymes. How do enzymes operate? So there are two hypotheses for the mechanism of action of enzymes. There is the lock and key model, and there is the induced fit model. So just clean the board and let me just show you that. All right, so this illustration here is showing the first hypothesis. It's called the induced fit model. Okay, so this is your enzyme, this is your substrate. I told you that enzymes will act on a substrate bar and they'll give rise to a product. Okay, so this is your enzyme, this is your substrate, and this part of the enzyme where the substrate will come to bind is called the active site. Take note, the specific part of the enzyme where the substrate will come to bind is called the active site. Okay, so now the induced fit model says that the substrate does not necessarily have to, have the shape of this active side of the enzyme. That once the substrate comes to the enzyme, the enzyme would adjust the shape slightly to fit this substrate. So you can see it's illustrated here that the substrate has a different, like a zigzag kind of shape, which does not match this active site here. But when it comes in, there will be an adjustment, a slight change in the shape that will now allow the substance to bind. That's the first model. That's the first hypothesis of how enzymes, work. That's the first one. All right, so this is the second hypothesis under the mechanism of action of enzymes. It's called the lock and key model. How this model, what this hypothesis says is that the substrate must have a shape that corresponds with the active site of the enzyme before the enzyme can act on it. That the substrate must have a shape that corresponds to the active site of an enzyme before the enzyme can actually act on it, just like you are seeing in the illustration here. So see that the shape corresponds to this active site shape as well. You get, that's when you can now have an enzyme substrate complex and the reaction can be carried out. So it's different from that induced fit model that says that it does not really matter, that the enzyme will just adjust. You get, so that's the mechanism of action of enzymes. Okay, so moving on, we'll talk about some terms that confuse students a lot in enzymes, this topic, enzymes. So it's not difficult at all. What are apoenzymes? What are cofactors, holoenzymes, coenzymes, prosthetic groups? Okay, so here's what it means. There are some enzymes that are like senior enzymes. They cannot work until there's a cofactor attached to them. Like they cannot be catalytically active. You get, they cannot carry out their enzymatic function until there's something attached to them called a cofactor. So that enzyme without the cofactor is just a protein. So it's called an apoenzyme. You get, the protein part is just called an apoenzyme. But once a cofactor comes and attaches to it to make it complete, it now becomes a whole enzyme called the holoenzyme. You get, so a holoenzyme is a complete enzyme that has both the protein parts called the apoenzyme and the non-protein parts called the cofactor. Do you understand? So the protein part is just the apoenzyme, but when the cofactor comes, it now becomes a holoenzyme, right? So there are two kinds of cofactors, right? One is called the coenzyme, the other is called the prosthetic group. The coenzyme is, okay, there are two types, and one is not tightly bound to the enzyme, and that one is called the coenzyme. So it's sometimes, most times, coenzymes are not necessary, are not mandatory for an enzyme to work. But when you're talking about the prosthetic group is one that is tightly bound to the apoenzyme. You get, and in that case, it is very mandatory for its function. All right. So I'll just reveal what I just said now. That there are some enzymes that are complex enzymes. They cannot work until there is something attached to it. So if that thing is attached, it's now called an holoenzyme. But without that thing, it's called cofactor. You get, so without the cofactor, it's just an apoenzyme, just like a protein part alone, just an apoenzyme. You get, but when the cofactor now comes, it now becomes a holoenzyme. So now I see that there are two kinds of cofactors. There's one that can be loosely bound, that is not very mandatory for the enzyme to carry this function, and then there's one that is tightly bound, that is now mandatory. The one that is loosely bound is called the coenzyme. The one that is tightly bound is called the prosthetic group. Right. Okay, so lastly for this video, we'll talk about some factors that affect enzyme activity, and it's pretty straightforward. The first is temperature. Temperature, usually an increase in temperature will actually facilitate enzyme activity. Yeah. The second is pH. pH for different optimal pH or optimum pH for different enzymes varies. So there's some enzyme that prefers to work in particular pH. So that one is enzyme dependent, but pH actually affects the activity of enzymes. Next one is what enzyme concentration. So when there are plenty of the same enzymes in a particular place, then the function of that enzyme can be carried out better, obviously, but it's straightforward. The fourth is the substrate concentration. So if there are more substrates in a particular place than enzymes, then the enzyme activities cannot be very, very efficient. You get, so that's understandable. Then presence of inhibitors. So anything that inhibits an enzyme activity, if it's present, the enzyme activity can be affected negatively. Then presence of activators like metal ions. So if there's anything that activates enzyme activity like metal ions, and that there, I mean, enzyme activities can be affected positively. So it's very straightforward. So in the next video we'll talk about the classification of enzymes. Is very important, the standard classification of enzymes. So we'll just do a separate video for that. So once again, if you haven't subscribed to the channel, I think you should do that right now, and then drop comments if you feel like there's a question you have. Maybe I was too fast or anything, just let me know in the comment section. You want me to teach a different topic, just let me know. Just drop comments. You can reach me on WhatsApp. I think I'll just drop my number on WhatsApp on one of the comments in the comments. And I think that'll be it. So just watch out for the next video, right? Like, subscribe, share.